Hello, everyone, and welcome to today's live broadcast, The High Resolution Solution, Accurate Mass for the Clinical Research Laboratory, presented by Natalie Rasmussen. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by Agilent Technologies. Agilent is a leader in life sciences, diagnostics, and applied chemical markets. The company provides laboratories worldwide with instruments, services, consumables, applications, and expertise, enabling customers to gain the insights they seek. Agilent's expertise and trusted collaboration give them the highest confidence in our solutions. For more information, please go to www.agilent.com. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce Natalie Rasmussen. Natalie grew up in Salt Lake City, Utah area and earned a BS in biology from the University of Utah in 2004. She began working in the toxicology area at ARUP Laboratories in 2005, where she prepared samples and analyzed them via immunoassay and mass spectrometry. Natalie accepted a position as Research and Development Specialist at ARUP's Institute for Clinical and Experimental Pathology in 2012, where she primarily works on time-of-flight mass spec-based testing. She also has some experience with immunoassay and MALDI-TOF. In her free time, she enjoys exploring the playground that is Utah by hiking and camping. She also enjoys reading, cooking, playing softball, and attending sporting events, especially at the University of Utah. Please join me in welcoming Natalie Rasmussen. I will now turn the presentation over to her.
Thank you for that introduction, and hello everybody. The title of my talk is The High Resolution Solution, Accurate Mass for the Clinical Research Laboratory. So some learning objectives for today. Hopefully by the end of my talk, you will be able to describe how time-of-flight mass spectrometry works, compare and contrast TOF-MS with LC-MS, and begin to assess what you will need in order to integrate TOF-MS into your laboratory. Here's a brief outline of my talk. First, we will discuss what accurate mass is and what mass resolution is. We'll talk a little bit about time-of-flight mass spec anatomy. I'll talk about method development, some interesting results in data mining, the strengths and weaknesses of the instrument, some future directions, and then I will open it up for questions. Let's start off with a little chemistry review. Individual atoms are too small to put on a scale and weigh, so mass is based on the carbon-12 scale. The atomic masses of all of the elements are based on how heavy they are relative to carbon-12, which weighs exactly 12 AMU. The mass listed on the periodic table, upper right-hand corner of this example, is the average atomic mass. It is a weighted average of the element and its isotopes. In high-resolution mass spectrometry, we use exact mass, not the average mass, when calculating the weight of an analyte because the instrument is capable of detecting all of the isotopes individually. The big number in the upper left-hand corner is the atomic number, which corresponds with the number of protons in the atom. Exact mass, like we just talked about in the previous slide, is calculated based on chemical formula. accurate mass is what the instrument measures. And then the atomic mass is determined for each element relative to carbon with the exact mass of 12. There are two types of resolution when it comes to mass spectrometry: chromatographic resolution and mass resolution. We will focus on the latter one today. Mass resolution is the ability to distinguish two ions of slightly different mass over charge in a mass spectrum. The narrower the peak, the higher the resolution. The higher the resolution, the higher the mass accuracy, which means the better ability for the instrument to be able to separate two compounds very close in mass. So the formula is resolution equals mass over the delta M, which is full width half mass. Or an older definition is mass over, is the average of the two masses over the mass peak separation. So the same conclusion can be reached, however. So a bigger R means a smaller mass difference can be resolved. So what does that mean? So a triple quad or a single quad is a nominal mass instrument, and a TOF or a QTOF is an accurate mass instrument. On a nominal mass instrument like a triple quad, the peaks of the two analytes aren't very sharp, and there's very little baseline resolution. The analytes look like they're about 614-ish and 615-ish, but on a high-resolution instrument, the peaks are very clearly separated, and the mass is measured out to thousandths of a Dalton. When the resolution equation is applied, the triple quad has a resolution of about 903, and the TOF or the QTOF has a resolution of over 14,000. The peak is much sharper on a high-resolution instrument, and you can see the big space in between in the TOF example versus the triple quad example. Let me give you a brief overview of what's going on inside the instrument. The LC stream enters the source, desolvation occurs, and ions are sucked through the capillary. They go through an octopole and a series of lenses and a quadrupole. If you have a QTOF, ions will flow through the collision cell. Ions are fragmented, and the beam of fragments, or intact ions, if you are using a TOF instrument or running the TOF or the QTOF in TOF mode, are further compressed and shaped before passing into the flight tube portion of the instrument. The pulsar assembly is a stack of plates with a hole in the middle. The bottom plate, called the pusher, sends ions up through the plates and toward the top of the flight tube with an electrical pulse. The reflectron, or ion mirror, sends the ions back down toward the detector. So how is all of this resolution achieved? Physics, but only the algebra-based kind, thankfully. All charged ions, Z, have a potential energy proportional to the acceleration voltage. When the ion beam enters the pulsar assembly of the flight tube, potential energy is converted into kinetic energy. The first law of thermodynamics states that energy has to be conserved, so potential energy equals kinetic energy. Velocity is the distance, or the length of the flight path, divided by time, so the time from the pulsar to the detector. So from all of the stuff we haven't thought about since our second year of college, we can derive the main TOF equation. The time of flight equals a constant times the square root of the mass over charge, or simply put, the heavier the ion, the longer the flight time. The instrument is calibrated with a range of masses, and the unknowns being analyzed are compared to this calibration curve. Based on their time of flight, the mass can be determined. Shifting gears a little bit now that everyone is familiar with how the TOF works, let's talk a little bit about method development. It's pretty easy for the most part. When I'm developing a new method, I start by creating a database of the analytes that I'm interested in. The chromatographic part of the method development is much like it is on a single quad or a triple quad instrument. After I figure out what columns and mobile phases I want to try, I prepare a solution of the analytes I'm looking for. Things get a little bit different when it comes to injecting the standard solution, however. I typically prepare a solution containing all of the analytes and also two solutions separating any isobaric compounds. Since the TOF is basing identification on exact mass only and not fragments, we have to rely on retention time to correctly identify any isobaric pairs. Having them in separate solutions allows you to figure out what time each one elutes. And then I try to answer the following questions when I'm reviewing data. Did I find everything? Well, hopefully I did. If I missed something, there's ways to dig deeper and figure out why, like looking at the extracted ion chromatogram. Usually, if something is missed, it's because two analytes are interfering with one another or an isobaric pair is co-eluting. I ask myself, what is co-eluting and does it matter? In my experience, generally compounds can co-elute as long as they are far enough apart in mass to not interfere with one another's isotopes, which is part of the source identification algorithm, or one isn't going to completely mask another because it's going to be highly concentrated in a biological matrix like urine. Are isobaric pairs fully resolved? There are none in this example, but if there were, they would need to be at least baseline resolved and preferably have a little space in between them. So when your medical director asks you to do something else after your chromatography is all worked out, this is probably the face you give them. But actually, it's really easy to add a compound after your method is developed. You add the analyte to your database, inject a standard to get the retention time, and then add it to your stock solution to make sure it plays nicely with everything else. Isn't life nice when you don't have to optimize or de-optimize individual compounds or look for transitions? Once you have your LC method mostly dialed in, you can start playing with the mass spec parameters to get the best signal. Some parameters will depend on the flow rate of your method, like gas temperatures, nebulizer pressure, and acquisition rate. If your chromatography is really fast, the instrument needs to scan faster in order to ensure that it finds enough points across the peak. The higher the scan speed, though, the lower the sensitivity. So there's a sweet spot that needs to be found. Voltages can be molecule dependent. So delicate molecules like amphetamines will fragment in the source if you hit them with something too hard. But a compound like THC glucuronide needs a little extra kick so we can apply a nozzle voltage. Different time segments can have different properties, which is useful for methods that have a lot of analytes. And you can also apply a divert valve at different time segments, and you can even switch polarity at different time segments as well. But as we know, nothing is ever as simple as it seems. If we could only live in a world where we only looked at standard solutions. We've learned a few things along the way in our TOF journey here at ARUP. What other common things in your method are isobaric with compounds that you're looking for? Desvenlafaxine is an active metabolite of venlafaxine, an SSNRI called Effexor, and it's also prescribed alone as Prestique. Years ago, not too long after we got our first TOFs, we started seeing tramadol with a slight retention time shift and no metabolites, and this is unusual in urine. After a little investigative Googling and looking at the forensic toxicology database from Agilent, we discovered that this could be desvenlafaxine interfering. So we went and mined the data and looked for the parent drug, venlafaxine, and then ordered standards to verify the retention times of the interfering drug and its parent. We were looking at marijuana metabolites in umbilical cord and meconium for a research project when one of our scientists discovered that progesterone and cannabidiol are isobaric. They eluted far enough apart not to cause any problems, luckily. Compounds that co-elute and are 1 to 2 AMUs apart will likely have isotopic interferences. Since isotope ratio and pattern are part of the identification algorithm, they really need to be separated if possible. This includes internal standards as well, so we try not to use the D3 unless we don't have any other option. So the TOF sees everything. Since you're collecting full scan data, it's going to pick up everything you put into it. So here are a few pearls of wisdom. Being clean and testing everything before you put it into use is paramount and will save you the heartache and pain that comes with troubleshooting. Do not use parafilm, ever. It's the saran wrap of the laboratory, and we love it, but the TOF does not. It will show up in your samples, it'll show up in your instrument, and it's a mess to sort out. Only use non-leaching plastics if you must use plastic at all. Some types of gloves can even cause problems. The highest quality solvents need to be used on board the instrument. We use Optima-grade methanol and acetonitrile and Millicue water in our laboratory. We've tested the lower grade stuff and it just causes problems. Hand wash your dishes. Detergents will show up in this signal just like parafilm will. We've found that it's best to keep a separate set of glassware for TOF-related preparation. Just rinse it with some IPA and methanol to keep it clean. If it's really dirty, a light acid wash followed by a thorough rinsing with IPA and methanol will clean things back up again. Anytime you get a new lot of anything that touches your specimen or the instrument, test it to make sure that it performs like the previous lot and doesn't have any interferences. I know this is standard practice, but it's especially important to remember for an instrument that sees just about everything you put in it. After all of this, you will still have interferences you can't explain from time to time. Biological matrices are complex, and people have all sorts of diseases and take all sorts of medications that could cause something to show up that you didn't anticipate. If you start off with a good, clean sample prep and instrument practices, there's less of a chance of something getting weird and causing problems. This is an example of polyethylene glycol contamination. See the fin pattern? This is characteristic of any sort of plastic contamination. PEG is 44 AMU parts, so it's usually pretty easy to diagnose. And when we see this, we typically have to flush our LCs out and clean everything really thoroughly, and it seems to clear the problem up. The instrument itself is pretty easy to maintain from day to day. You do the normal LC stuff like checking the solvent levels and priming the pumps. Wipe the inside of the spray chamber down with a mixture of IPA and water, and then just do a mass calibration for the instrument. All in all, it takes less than 15 minutes. Major service, like a front-end cleaning, will depend on what and how much you're running. We are a busy lab. Thousands of samples per month pass through these instruments. What we found is that every instrument has its own personality of sorts. Some can go longer than others without being cleaned. It usually takes 3 to 7 days for an instrument to come back to normal post-cleaning. The key is paying attention to changes in your system and being proactive. Having a set of criteria when have a set of criteria for when your instrument needs service. Area counts are the biggest indicator. We are fortunate because we have a factory-trained service group of engineers in-house because we have over 100 mass specs of all shapes and sizes and brands in the building that they are capable of fixing and maintaining. So when one goes down, help is not far away. But like I said before, the key is paying attention to changes in the system and acting on them before things become a crisis. Set criteria that meet your needs and get help before it blows up in your face. Since we're collecting full scan data, the instrument generates a ton of data. So files can be greater than 1 gigabyte per sample. Data storage is a big issue. We max out hard drives in about 40 days. Once a hard drive is full, the instrument won't run. So you'll need to have a data storage strategy in place, specifically if you're a CLIA lab, since some sort of archiving is required. We archive PDFs of reports, and the actual mass spec data is deleted after a set period of time. You'll have to figure out what works best for you, but keep in mind that a 2 terabyte hard drive is going to fill up faster than you think. Now I want to talk to you about some of the ways in which we use time of flight in our laboratories. This is a method used for testing for drugs commonly prescribed in pain management programs. These drugs are prone to misuse or diversion, so doctors want to make sure that people are taking what they've been prescribed and aren't enhancing their prescription therapy with anything else. This is 35 drugs and metabolites and 7 internal standards in about 3.5 minutes. I presented this particular method on a poster at SOFT in Atlanta, Georgia last year. This method is run on our Agilent 1260 HPLC coupled to an Agilent 6230 time of flight mass spectrometer. Chromatographic separation is achieved on a Phenomenex Kinetex F5 column heated to 40 degrees, and the mobile phases are 5 millimolar ammonium formate, pH 3.5, and Optima-grade methanol. The flow rate is 1 ml per minute. The method uses an ESI source with a scan speed of 2 spectra per second. The gradient is pretty simple, and the whole thing is less than 4 minutes injection to injection. Sample prep to recover so many different drugs can be challenging. The summary on this page is a recent addition to the sample prep. We wanted to improve our detection of some of the benzodiazepines that are glucuronidated because the glucuronides don't recover well with our extraction method. So we had added a hydrolysis step. Rezorufin glucuronide is being used as a hydrolysis marker in each sample. So we take 200 microliters of our sample, controls, and calibrator, aliquot them out. We add a buffer, we add the glucuronide, the rezorufin glucuronide and beta-glucuronidase in the internal standards. We let it sit for 15 minutes at room temperature, and then we proceed with our normal extraction. And I put this slide out of order. This is courtesy of Lacey Taylor, who developed this method. This was just showing that our detection of the glucuronidated benzos was improved with the hydrolysis. So rezorufin was included to monitor enzymatic hydrolysis activity. Biological samples that included the rapid enzymatic hydrolysis step showed the presence of the rezorufin analyte as expected. Surprisingly, rezorufin analyte concentrations varied greatly in the biological samples, indicating the variability of extraction and enzymatic activity from specimen to specimen. Lacey Taylor is a newer addition to our R&D group, and she presented this at SOFT in Dallas a couple of weeks ago and actually won the Young Forensic Scientists Award for her poster, and so we're all very proud of her. So on to the rest of the sample prep. After the hydrolysis, samples are transferred onto a Biotage Isolute SLE plus 96 well plate. Positive pressure manifold is used to push the sample into the packing material. It's eluted with ethyl acetate and dried under nitrogen at 40 degrees for 10 minutes. We use the Biotage ACT Plate adapter because we learned that plates were getting contaminated when there were samples that had really high levels of things like methamphetamine or morphine, and we kind of hypothesized that it formed a cloud and would dissipate over the plate and contaminate wells around it. So this plate adapter seems to have eliminated the problem. This is a semi-quantitative assay. We're just calling things positive or negative, not putting a concentration level on it. So it's a 1-point calibration curve at the cutoff, and it's forced through 0. We're normalizing to 7 internal standards. We have QC at 50% and 150% of the cutoff, front and back. And qualitative results are based on the cutoff, mass match, retention time, and isotope spacing and abundance. Statistical analysis of our calibrator and control replicates are used to determine acceptance criteria for the mass match, relative retention time, and ion suppression in the internal standard. And last time I checked, our 50% control had a median CV of 11%, and our 150% control that contains all the analytes had a median CV of 9.2%. This is an example of the calibrator. Flagging calls attention to things that may be falling outside of the established criteria. You can see the calibrator with 2 QC points on either side in the bottom right-hand corner. And then in the bottom left-hand corner, you can see the stick chromatogram with boxes around each mass. And those are the isotope, that's for the isotope ratio and spacing, which I mentioned as part of the data acceptance criteria. So this report looks pretty complex, but really when you're reviewing it, the only things you have to look at are what is being flagged as outside of your criteria. Just some remarks on this particular test. Overall, it performs very well, but there's some challenges. The laboratory was confirming approximately 20% of samples by LC-MS. or 20% of the positive samples by LC-MS. Typically, this was because the metabolite pattern was abnormal or the score was suboptimal due to a high mass error or a slight shift in retention time, or what we call an isobaric imitator. So it's an uncharacterized interference, and we haven't been able to figure out what it is. This can push the turnaround time out a couple of days, and the sample has to be reprepped, and it's sent to another department in our building for confirmation. So obviously this is an ideal situation. Having a well-characterized assay with tight acceptance criteria and a good understanding of how things should look can help prevent these ambiguous results, but you're still going to have issues from time to time. This is an example of a biological specimen that needed to be confirmed by LC-MS. The mass accuracy was way off for both the alprazolam and its metabolite, alpha-hydroxyalprazolam, as is the match score. If you look at the isotope pattern, it is clear there's something interfering with the analyte. The specimen came back positive for both analytes, with alprazolam at 26 nanograms per mil and alpha-hydroxyalprazolam at 104 nanograms per mil. Since having this specimen confirmed on another platform in another laboratory is inefficient, we wondered if we could use our QTOF's fragmentation abilities to confirm the specimens instead. It would be cheaper because it doesn't require reprep by the other lab. I used the same chromatography and went from there. There are a few different ways to select what you want to fragment on the Agilent QTOF. I chose to use the Auto MS setting with a preferred list. By selecting preferred list only, the instrument will only fragment what is on that list, much like a triple quad. You can also have it fragment analytes from the preferred list first and then anything else after it if you're doing unknown screening, if it has time to do it, or it can fragment everything that meets a criteria that you set. So you can set like an ion or an abundance threshold. Collision energies in this particular method were set at 10, 20, and 40. The acquisition rate for the mass spec was at 8 spectra per second, and for MS it's set to 4 spectra per second. By making use of the collision cell, we can add a fragmentation pattern to the other acceptance criteria of mass accuracy, isotope fidelity, and retention time to verify positivity in these specimens with suboptimal results on the TOF. I presented this method at SOFT in Dallas this year. This method is not currently in use, however. We have the 6550 model of the QTOF, which is more of a research-grade instrument with an overprotective research scientist, me, at the helm. It didn't fit well into the laboratory's workflow, so they continued to confirm what they needed to by triple quad, but I was able to demonstrate that should they want to go down this route, they could use this instrument to confirm instead of the triple quad. This is what the QTOF data looks like in MassHunter Qualitative Analysis. The diamonds across the peak in the chromatogram results plane are where the instrument has fragmented. In the MS spectrum results pane, at the top box shows the intact ion, and the boxes below show the fragmentation pattern at each collision energy. This pattern is compared to the fragmentation pattern in the library and is assigned a match score in addition to the overall score that includes mass match, isotope fidelity, and retention time. This is a comprehensive drug screen developed on the TOF by Catherine Smith and was presented at MSACL earlier this year. It's meant to be an overdose panel, so we have really high cutoffs and long, beautiful, well-separated chromatography. Sample preparation is a dilute and shoot approach, with the diluent being an internal standard solution. The separation is achieved on an Agilent pour shell EC C18 column, and mobile phases are 5 millimolar ammonium formate and Optima grade methanol in positive mode. The reference masses, which are infused along with the samples, can be used to evaluate ion suppression since they are constantly being infused to correct the mass calibration. Both masses are fairly smooth all the way across the chromatogram, indicating that despite being a dilute and shoot method, there is little to no ion suppression. Using the divert valve is critical with the dilute and shoot prep because all of the junk comes off the column at the beginning and at the end, and so we don't want to send that into the mass spec. So that's what those lines on either side of the mass of the chromatogram are the 1 and 2 and 2 and 3. Those are different experiments. So experiment 1 is being sent to waste and experiment 3 is being sent to waste. So we're only collecting between experiment 2. And this is what negative mode looks like. Separation is done on a shorter C18 column with a bit slower flow rate than the positive mode method. The aqueous mobile phase is 0.1% acetic acid in water and optimum ethanol. Using the acetic acid rather than the ammonium formate and formic acid mobile phase used in positive mode boosted our sensitivity quite a bit. MassHunter Qualitative Analysis is used to evaluate the data. Like in the first method I discussed, a 1-point calibration at the cutoff forced through the origin is used. 2 levels of QC on either side of the calibrator and a negative controller used to evaluate the calibration. This method is not meant to produce a quantitative result, but rather a present or not present result. Acceptance criteria are very straightforward. If an analyte doesn't meet them, it is considered not present. So I have a confession. I am the world's most impatient chromatographer. A little over a year ago, I was talking with one of our medical directors, Dr. Fred Strathman, about other ways in which we could use the TOF in the laboratory. We began to wonder if we could use the TOF to replace enzyme immunoassay portion of a screen with reflex approach to drug screening. Enzyme immunoassay is plagued with issues like false positives and false negatives. For example, well-butrin and pseudoephedrine can cause an amphetamine immunoassay to be positive. Clonazepam and 7-aminoclonazepam, on the other hand, have extremely poor cross-reactivity, like they need to be 1,000 nanograms per mil in concentration in urine to show positive. So they are likely being missed. Enzyme immunoassay kits are also expensive, which is another disadvantage to the technique. Don't get me wrong, immunoassay is a good broad test and it is robust and it has served laboratories well for years. We just wanted to see if we could do better. So the goals of this project were to make the screen faster, cheaper, and have a higher specificity and sensitivity than the immunoassay. Cutoffs would be analyte specific rather than drug class specific. Another advantage to screening by TOF is we would not need to wait for an immunoassay kit to be developed for a new drug that's on the market. We could just get a standard for it and add it to the assay. Since any positives would still be reflexed to confirmation and quantitation by LC-MS, the chromatography didn't need to be perfect, but just good enough. Sample prep is dilute and shoot. This is 76 compounds in positive mode in less than 90 seconds. A short column, high temperature, and fast flow rate are used. I don't want to give away too many details, though, because I haven't published this method yet, so this is just a preview. This contains all of the typical drugs of abuse like amphetamines, cocaine, opiates, benzodiazepines, and PCP, as well as nicotine and its metabolites, and some tricyclic antidepressants. This is negative mode, 20 analytes in less than 90 seconds, including ethyl glucuronide and ethyl sulfate. Also included are THC, barbiturates, and some of the benzoglucuronides. Pretty cool, right? But what about creatinine? We did that too. So I've got a 5-point calibration curve for creatinine and 4 levels of QC, and we are just a few experiments. We just have a few more experiments to run, and we'll be ready to write the manuscript and hopefully present this at a conference soon. Time of flight is a very powerful tool when it comes to unknown screening. A biological specimen was suspected of having some sort of amphetamine-like compound in it. My medical director gave it to me and said, "Here you go, have fun." So I love troubleshooting and solving mysteries, so I happily ran off to the laboratory to work on the QTOF. So I diluted the specimen in mobile phase at a ×20 dilution, and then I analyzed it using kind of a generic method, probably a C18 column. I used MassHunter Qualitative Analysis, Find by Formula, and the Agilent Forensic Toxicology PCDL, and the most likely hit I came up with was this 2,5-dimethoxy-4-chloramphetamine, or DOC. It had a score of 98 with mass match and isotope fidelity, but I didn't have a retention time because I didn't have a reference standard. Since I didn't have a reference standard to verify the retention time, I made an auto MS method on the QTOF, knowing that the fragmentation would give me another level of certainty. When comparing the fragmentation pattern to the library, there was a match score of 95. So I had a 98 on the first match score and a 95 on the library score. So do we even need to order a reference standard? It's an interesting question to think about. If you have an excellent score for mass match, isotope fidelity, and fragmentation pattern, what are the chances the software is telling you wrong? Well, I don't know the answer to that exact question, but we were still quite confident in the data, but I went ahead and ordered the DOC anyway, and we could verify the retention time. And as you can see in this example, the reference standard and the drug and the biological specimen matched. So it's very likely that this specimen did contain the DOC. TOF is a powerful tool with a lot of applications. You can do targeted screening. You can do untargeted screening. It's very user-friendly. Method development is generally easy, and new compounds can be added quickly. But assays need to be well-characterized. You need to make sure that you have strict acceptance criteria and have tried to account for any possible isobaric interferences that are not part of your assay. If anyone is interested, any of the presented published methods I talked about, I can get the posters to you. I would like to thank ARUP Laboratories, Dr. Fred Strassman, Catherine Smith, Lacey Taylor, the ARUP Institute for Clinical and Experimental Pathology, and the Clinical Toxicology 3 Laboratory. I would like to also thank Agilent Technologies, specifically Carrie Adler, one of my former ARUP colleagues. John Palmer and Julie Ciccelli are excellent resources when it comes to chromatographic separation and QTOF help. And then I would also like to thank LabRoots for giving me the opportunity to talk to you guys today. And with that, I would like to open it up for questions.
It's time for Q&A. Thank you, Natalie, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask Natalie, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click the send button. We will answer as many of your questions as we have time for. Let's get started. Natalie, our first question is, how is the match score calculated?
It's an algorithm in the software. It takes into account mass accuracy, retention time match, and isotope fidelity, so isotope ratio and spacing. And then that can also be changed. The weighting can be changed depending on the type of analysis you're doing. So if it's a fast chromatography, you can decrease, for example, the weighting of the isotope fidelity and increase the weighting of the other two parameters.
Now, what sort of experience does a tech need to competently run the instrument and analyze the data?
I started running this instrument as a med tech on the bench, and having a little bit of a mass spectrometry background helped. It's certainly something that, you know, a bachelor's level like medical technologist can run and understand. It's not extraordinarily difficult to learn how to analyze the data. You just have to know what good data looks like and have that well defined.
Very good. Now, let's see. Here's another question. Nellie, when conducting your general unknown screen with the master PCDL, did you encounter erroneous results by using only accurate mass? If so, how did you determine what is real versus erroneous?
I didn't have any erroneous results, but that could I think as far as erroneous results, if you had like a bunch of matches for a bunch of isobaric compounds, you would have to either fragment using the QTOF or order standards and match to retention time. So yeah, I guess that's probably how I would approach it.
Very good. Let's see. Let's give the audience just a moment to see if we have any more questions coming in. It looks like, let's see. As we're waiting for any questions, Natalie, do you want to give us any final comments for the audience? Anything you'd like to add?
Nothing comes to mind right now. I think I covered almost everything I wanted to, but I'm always happy, like if anybody thinks of a question that they want to ask after this, they can always send me an email, or you guys can probably tell them how to get in contact with me, and I'm always happy to answer stuff offline as well.
Yes, absolutely, because today's webcast will be available for on-demand viewing all the way through February 2017. And during that demand viewing, you can submit questions, and Natalie would be happy to respond to you offline. And you will be receiving an email from LabRoots letting you know when this webcast will be available for replay. And we do ask that you share your announcement with your colleagues who may have missed today's live event. Let's see. I believe we have time for another question. Nellie, with such short run times, did you have a post time to help equilibrate before the next injection?
Yeah, my post time, let's see, I think the post time on the first example with the 35 drugs is around 1 minute. It's 30 seconds in the rapid screen, and then I'm not sure what the post time is on the longer comprehensive method.
Very good. Let's see, I believe those are all the questions that we have. Again, you're welcome to submit during the on-demand, and Natalie will respond offline. And again, it's available through on-demand viewing through February 2017. And again, you'll receive an email from LabRoots letting you know when this webcast will be available. Thank you for joining us. I believe that's all for now. We hope to see you again soon. Goodbye.